So today we're going to talk about human metabolism. So everything that happens once all the nutrients are absorbed after the digestion process. And the first thing we need to know is a couple of organs that are critically important for evaluating nutrients after absorption. And the first one, of course, is the liver. So the liver is strategically positioned because it will absorb all the nutrients that actually come in after the digestion process. So here you can see actually how it looks like. So here we have the small intestine, all the water-soluble nutrients. So these are proteins, amino acids in this situation, and carbohydrates, monosaccharides. They enter here in the portal vein and they go directly to the liver. So the liver will actually control these types of nutrients before they go to the rest of the organism. For everything that is fat soluble, it will go to the lymphatic tissue and it will have a bypass of the liver. So it will not go to the liver, otherwise you get liver fattening, and it goes directly into the adipose tissue. The liver is also the detoxifying organ, so all potential toxins will first go to the liver and will there be detoxified before they can go to the rest of the human body. We also need the brains in our discussion. The brains contain a blood-brain barrier, which is not accessible for lipids. So the only molecule that can penetrate quite well through the barrier is glucose. And the brains will actually normally function exclusively with glucose. But as soon as glucose is vanished or is depleted, the alternative is the use of ketone bodies, like acetone, which is actually a sort of an emergency situation, but it is more and more used also today in, for example, ketogenic diets. So diets where the carb loading is very low, so low intake of sugars. In this case, the body is actually forced to use more lipids, and these lipids will actually create so-called ketone bodies, which are also a substrate for the brains to work and to generate energy. It's not an ideal situation. You don't have to maintain the situation for a long period of time, but it is an alternative in case glucose is absent. In the muscles, we have anaerobic and aerobic fibers. Aerobic fibers are oxidative, so contain oxygen. These are the red fibers. The white fibers are anaerobic, do not contain any oxygen. And there's a specific substrate, which is called creatine phosphate, which is used for production of ATP in the muscles. In the lipids, so in the fats, we have adipocytes, so fat-containing cells, which can either be brown or white. Brown cells are not depositing lipids. So in this case, they are depositing lipids, but they are not really producing ATP, while white adipocytes can use lipids to produce ATP. And in infants, we mainly found brown adipocytes. In adults, we mainly have white adipocytes. So you can think a little bit yourself why infants mainly have brown adipocytes and why this is changing into white adipocytes when people are getting older. And there's a specific metabolic function for that. And then we have the kidneys. They contain a cortex, which is aerobic outside, and the inside is the medulla, which is anaerobic. And of course, aerobic or anaerobic also determines the substrate and the type of metabolism. The whole system is regulated, and this is done by the use of hormones. The best well-known hormones are glucagon and insulin. Glucagon is actually releasing energy. Insulin is storing energy. So it means that if there's a lot of insulin there, then technically speaking, the lipids are not well used. They are mainly stored. If insulin levels are low, then glucagon is high, In those cases, lipid reserves can much better be metabolized and can be used to generate energy. So all of these are produced in the pancreas, and the proportion between insulin and glucagon is the ratio which is normally used in normal metabolism. The whole system is made more complex by the use of hormones like catecholamines. These are so-called stress hormones like adrenaline and noradrenaline, and they are actually also influencing the whole process. For example, if you're under stress, you can mobilize much better your lipid reserves, which is actually stimulated by these stress hormones. And the hormone which is responsible for the release of lipids is called hormone-sensitive lipase. It's a lipase that you can find within the adipocyte. It works in the same way as the lipase in the GI tract, but it is hormone sensitive. So it is influenced, for example, by stress hormones, but also by insulin. So insulin can actually reduce the activity of hormone-sensitive lipase, meaning that if there is a lot of insulin, so in other words, there's a lot of sugar, then the lipids in the adipose tissue will not be released. They will be stored. While if you have a lot of stress, then the hormone-sensitive lipase is active, it's stimulated. You have more release of lipids from the adipose tissue, and this will create more substrate to generate energy. So let's start with the carbohydrates. So if we look to an ordinary blood glucose level as a function of time for humans, then you can see that the fluctuation for glucose levels is not really high. There's a small change as a function of time, but generally speaking, it's quite constant at the level of 4 to 6 millimole per liter. However, the fluctuation in insulin level is much higher. So you can see here the fluctuation in the morning when you're taking breakfast, fluctuation around lunchtime, and the fluctuation in the evening. And during the night, insulin levels gradually drop, while glucose is still kept rather constant. And in the morning, the whole process starts again. Glucose has to stay constant because it is the source of energy for the brain. So if you want to maintain energy supply to the brain, you have to maintain glucose levels in the blood as a function of time. Carbohydrate metabolism on a general basis is a simple metabolism because everything is transformed into glucose. You may have galactose or fructose or whatever, but more or less after a while in the body it is converted to glucose and used as such. The blood glucose has a dynamic equilibrium around 5 millimole per liter, we already explained. If it goes below 3 or above 11, you get dysfunction. Below 3, you can actually faint because you don't have enough supply of glucose to the brains. If it's higher than 11 and maintained for a long period of time, then you come into a situation of diabetes, whereby you have too high levels of blood glucose. If we look into biochemistry, there are in fact 4 different situations that are interesting in terms of carbohydrate metabolism. The first one is that in the muscle, there is no glucose 6-phosphatase. If you look into your biochemistry, you will see that glucose 6-phosphatase is the last enzyme before glucose can be converted into free glucose. So if you start from starch and you break it down, you end up in glucose, provided that there is glucose 6-phosphatase. And this glucose 6-phosphatase is not present in the muscle. So it means that the muscle, who has glycogen, which is a type of starch, it can break it down, but it cannot break it down to glucose. It can only break it down to glucose 6-phosphatase. And this one can then be used to generate energy for the muscle. But if glucose 6-phosphatase would physically be present, then glucose is produced, not glucose 6-phosphatase, and the glucose is free to diffuse into the blood, and it could be used in those situations by other organs. So since the muscle does not have this enzyme, it means that the muscle will never be able to supply glucose to the rest of the body. So if you're short in glucose and the brains need glucose, you cannot get it from the muscle because of the lack of that enzyme. The liver can. So the liver has glucose 6-phosphatase. So in case there is a shortage of glucose for the brains, then the liver is capable of supplying extra glucose to the brains. The second conclusion for carbohydrate metabolism is that glucose synthesis from fat is not possible, and that there is a limited fat synthesis from glucose. For both of these conclusions, I will refer you to the problem-based learning questions of Chapter 4. I will come back to that in a minute. And finally, in the urine, you don't have a large loss of glucose. If there is glucose in the urine, it probably means that you are somewhere here in a situation of diabetes, because there is too high accumulation of glucose in the blood. It cannot go into the cells, so the chances are bigger that it will be recovered in the urine. And you can do a test in the urine to see if no glucose is there. So you already understand that the body will do everything which is possible to keep the blood glucose concentration constant. And it can do that by glycogenesis, which actually means that glucose can go into glycogen. That's the storage organ for glucose, which is done in the liver and the muscle. In the other way around, so supplying glucose to the blood can only be done by the liver. It's called glycogenolysis. The muscle will not do that because it lacks this final enzyme. The second important function is gluconeogenesis, which means production of glucose coming from substrates different than glucose or sugars. For example, amino acids, or glycerol, or lactic acid. These types of substrates can be converted into glucose by the use of the liver. And then blood glucose can partly be converted into adipose tissue, but probably it will not be that large. Part of it is fatty acids, and part of it is the glycerol that can also be used for gluconeogenesis. So you can see you have different options, and it's not so easy to be completely short of glucose. But if it happens, then you need to interfere as quickly as possible by supplying glucose by the food. If we want to influence the process, an option we have is the so-called glycemic index. A glycemic index is determined by measuring the increase of blood glucose as a function of time. So you get an area under the curve by just asking people to take blood from time to time after consuming a standard meal. And then you just measure blood glucose as a function of time, and you measure the area under the curve. You relatively express this area to a standard. And the standard is usually glucose or white bread. So people are consuming either pure glucose or pure white bread. They measure the area under the curve, and this area under standard conditions is actually normalizing the area for your intervention conditions. They do that because it seems that for the same doses of glucose or white bread, individual people will respond in a different way. Some of them will reabsorb fast, some others will reabsorb a little bit slower. So in order to correct for that, you standardize it by dividing it relatively to the standard. And so you have sugars that have a high glycemic index or a low glycemic index. High is, for example, if you have a fast digestion, like in the case of sport foods, for example, you want the athlete to have very fast supply of energy, then you need to have it absorbed very fast. That's a high glycemic index food. Slow glycemic or low glycemic index foods are foods where the carbohydrates are much slower digested and the release in the blood is also delayed. And so in this case, you have a low glycemic index food. Monosaccharides are fast or high glycemic index loads, and slowly digestible starch, so starch which is a little bit modified or which is not well cooked, for example, or a raw potato or whatever, you get much slower release of starch, and then you have a low glycemic index food. There are also carbohydrates who are non-glycemic, who do not have a direct influence on the insulin response. Dietary fiber, per definition, is not digested in the small intestine, but goes to the colon, so there is already a large delay in the digestion process. The final product is short-chain fatty acids, and some of these fatty acids, like propionic acid, can be absorbed and can be transported by the blood to the liver, and there it can also influence sugar metabolism. And so, but it's quite strongly delayed, and it doesn't give you the very high peak of glucose in the blood. So please try to remember what a glycemic index is, how we can use it to influence metabolism of sugars, and try to understand, based on the pathways that you have seen in biochemistry, how sugars are normally metabolized in humans to generate energy. So let's look now at a number of practical situations. And the first situation is the so-called post-absorptive state. So it means that your last meal has already been absorbed for a while. And for example, you have eaten the day before, you go to bed and you sleep the whole night and you wake up in the morning, but you do not take breakfast yet. So the period just before breakfast is called the post-absorptive state. And in those situations, all the energy has to be derived from what is stored within the human body. So insulin levels are low because there's no sugar coming in. And in those situations, glycogen from the liver can be used to supply glucose to the blood. This glucose can go to the brain and it can be used for energy. Part of the glucose, in case still something is left, can go to the muscle, and in the muscle it can generate energy. From the muscle, you can produce alanine, lactate, or glycerol, which mainly comes from the adipose tissue. And these three substrates will be converted into glucose by so-called gluconeogenesis. And so the muscles are supplying the alanine, the medulla from the renal, so from the kidneys, as well as the blood cells will supply the lactate, and the glycerol is coming from the adipose tissue. In this situation, you get glucose mainly from endogenous reserves and from endogenous origin. What happens if you consume carbohydrates during breakfast? In those situations, a lot of carbohydrates come in. They are being absorbed after digestion. As soon as glucose ends up in the blood, there is release of insulin, which comes from the pancreas. As a result of that, glucose can be stored into glycogen. Glucose can go to the brain, and this glucose now is mainly coming from the food. The glucose can also go to the kidneys, to the muscles, as well as to the adipose tissue. And the insulin will follow, and the insulin will block the use of lipids. So for example, hormone-sensitive lipase is blocked. All the lipids will be stored in the adipose tissue. And here insulin is stimulating the use of glycogen and the use of carbohydrates by the muscles. So from this figure, you can already derive if, for example, you would be consuming a lot of sugar. For example, you are drinking 4 or 5 cups of Coke a day. And this regularly happens with several people that they're just consuming Coca-Cola. So in this case, a lot of glucose will end up in the blood. Insulin levels will always be quite high. And it means that simultaneously during the meals, if you consume lipids, these lipids will go to the adipose tissue and they will be stored because the insulin avoids that the lipids are directly used to generate energy. So the carbohydrates as such, in an indirect manner, can definitely contribute to supply and storage of lipids in the human body. This brings me to the first task, and this is for the groups A. And the tasks for A and for B will not be discussed in class. So you will have to make them yourself at home or with your group in case you want to discuss it. For 4A, the question is as follows. A person only eats potatoes. And potatoes are a rich source of carbohydrates, which is mainly there in the form of starch. So the person eats potatoes and a high amount of potatoes. So will it be possible to deposit more fat in the body coming from the potatoes? In other words, can carbohydrates from food be converted into body fat? Explain your answer on the basis of your knowledge of biochemistry. So you should think the potatoes are being broken down, they generate glucose, and you are consuming very high loads of glucose. Is it possible that the excess of glucose is used for the production of lipids? And if the answer is yes, how does it exactly work? If you want to do that in a quantitative manner, you can. For example, you can think about a person who is consuming more than a kilograms of sugar in a daytime. And just imagine that you are consuming 1 kilo of sugar a day, which is really a lot. I mean, you can do it. The experiment has been done, but you have to force yourself. And so nobody is consuming a kilogram of glucose a day. But if you force it, people can consume it. So the question is, if you would consume 1 kilogram of glucose, how much of that will be converted into lipids? Try to make a calculation and you will see. And so you will have to think what is actually going into normal maintenance metabolism, what is going into storage, and the remaining part will go into lipids. Yeah, so if you know more or less how much glycogen is stored in humans, how much glucose is needed for normal maintenance, then you also know how much glucose is left over, and that one can be used to convert into lipids. And so just make a small calculation on that. And then task 4B is for the second groups, which is just the same, but it's just the other way around. So in this case, we assume that the person does not contain any carbohydrates in the body. For example, he is doing a very heavy activity while he is not consuming any carbohydrates. So there is a depletion of glucose in the body. And the question is, what is going to happen now? So can lipids in the adipose tissue be used to supply extra carbohydrates, in this case in the form of glucose to the body? In other words, can the body convert lipids in the adipose tissue to glucose? Also here, explain your answer based on your knowledge of biochemistry. So you have to think a little bit. I start from the lipids. I will break them down into fatty acids. Where are these fatty acids going to? What is the breakdown product? In which cycle, biochemical cycle, will they appear? And will the cycle be functioning in a normal way or not when there are no carbohydrates present? Yeah. So it may be possible that you can feed the cycle, but it's not so sure that the cycle will stay operational as a function of time. And this is something you can try to figure out if you know more or less your biochemistry. So you should find a link between the glycolysis, the Krebs cycle, and the production or the release of lipids from the adipose tissue. You can go to the notes of the first lecture of this course, which is also online on Ch.4, where you can find all these cycles again in case you need them. Okay, I will stop the recording here because we end the first part on carbohydrates. So recording has started for the second part of the lecture, which will deal with fat metabolism. And fat metabolism is quite closely related to carbohydrate metabolism. In chapter 5, I will explain a little bit the energetic efficiency of both substrates. And there you will actually see that it is quite similar. So the body actually has a choice between either carbohydrates or either lipids. And from an energetic point of view, it doesn't make a big difference. The lipids themselves, if we look to their metabolism, then you will see that triglycerides are synthesized in the epithelium. I think we already discussed that last lecture, where I told you that after the digestion, the two monoglycerides and the free fatty acids are absorbed into the absorptive cell of the small intestinal wall. And there you get a resynthesis. So the triglycerides are produced again, and then they are transported to the lymphatic tissue. From there, they will go normally to the adipose tissue for storage. Once they are present in the adipose tissue, they can be released, and then they will be transported by the use of albumin. So albumin is a protein, it circulates in the blood, it can bind to what we call non-esterified fatty acids, the so-called nephas. And they are usually coming from the adipose tissue. So if the adipose tissue releases the nephas, they will bound to the albumin, and from there they can be transported for use as energy. A very big example or exception to that rule are the medium chain fatty acids. So medium chain means lipids that can be found between C8 and C12. So it's C8, C10, C12, these ones. If you go above that, for example, C14 or higher, then you have the long chain fatty acids. If you go below C8, for example, C4, C6, C2, C3, these are the short chain fatty acids. And the intermediate ones are the medium chain fatty acids. These fatty acids are not fully soluble in water, but they are better soluble in water than the long chain fatty acids. So for the MCTs, you do not need the normal digestion process that we discussed last week. So you don't have to go into the micelles, you don't have to go into the lymphatic tissue, which is the case with the long chain fatty acids, but not with the medium chain. The medium chain are sufficiently water soluble to also go to the blood and to be bound to albumin. So it's a sort of a bypass of the normal lymphatic system route. And this is interesting in terms of disorders of the fat metabolism. So if you have people that do not have an active lymphatic tissue, those people cannot consume fatty rich foods because the lymphatic tissue doesn't work. But they can consume medium chain fatty acids because they go to the blood, bind to albumin, and from there they can be used. So if you find nephas in the serum, which is a classical routine test that they can do in the clinics, you will see that the amount of nephas that are present in the serum are directly related to the release of lipids from the adipose tissue, from the activity of the hormone-sensitive lipase, and the rate by which nephas are used by the muscles. Yeah, so it's an important indicator to know whether or not fatty acid metabolism takes place. And short-chain fatty acids are well water soluble, so they don't need any carrier. They can directly be absorbed in the blood and be used for energy. Everything which is bigger than 12 carbons, so from 14 carbons onwards, need to be transported by so-called lipoproteins. And the lipoprotein is a particle that contains lipids on the inside, which is mainly the triglycerides and the cholesterol esters, which are all hydrophobic. So they are on the inside. On the outside, you can find phospholipids and you can find cholesterol, which are amphiphilic, so both water as well as fat soluble. And you also have a lipoprotein, which is called an apolipoprotein. It's a protein that can be linked to the lipoprotein, and this is the part that recognizes different types of organs. So if the lipoprotein needs to be absorbed in the liver, the particle will be recognized by the apolipoprotein that actually links up with an apolipoprotein from the liver. There are several types of lipoproteins, and it's a little bit technical, but in the clinical diagnosis world, it is very important. So the first one we have is the chylomicron, and the chylomicron contains 1% protein, 99% fat. The chylomicron will transport the lipids that come from the food. So they are produced in the wall of the GI tract. The second one is the VLDL. It stands for very low density lipoproteins. It's also called the pre-betas. And this one contains 7% protein, 93% lipids, and they are produced in the liver. So all the lipids that end up in the liver will be combined together into a new particle, which we call VLDL, and that one will also go to the adipose tissue. So the adipose tissue receives lipids from two sources. The first one are the chylomicrons, containing the lipids that come from the diet, and the second one is the VLDL, containing the lipids which come from the liver. Yeah, so the lipids coming from the diet do not go to the liver. There's a bypass using the lymphatic tissue. Then they end up in the blood and they go directly to the adipose tissue. The VLDLs collect all the lipids that are physically present in the liver and also sends them to the adipose tissue. This is an intermediate density lipoprotein, less important, but then two important particles are called LDL and HDL. LDL is low density lipoprotein and HDL is high density lipoprotein. You can see the compositions here. The most important thing is that these particles contain less lipids because the lipids are actually stored in the adipose tissue. You start from a chylomicron of LDL, you convert them into LDL or HDL by removal of the fat. So this is normally done near the adipose tissue. These particles are important because LDL is called the bad cholesterol. HDL is called the good cholesterol. So if people take samples of your blood and they analyze it, and they say you have a cholesterol problem, then it means that your LDL is too high and your HDL is too low. If the cholesterol levels in the blood are fine, then you have high levels of HDL and low levels of LDL. Why is this important? Well, these two particles play a role in development of cardiovascular disease in humans. It seems that the fatty streaks that are deposited near the wall of the arteries is mainly caused by LDL. and not by the normal LDL, but by the oxidized LDL. So the lipids within LDL are oxidized. This particle will deposit near the wall of the aortas and will induce, after years of intervention, it will induce cardiovascular disease. While the HDL is a positive particle because it will take away the cholesterol and the lipids from the arteries back to the liver. So the more HDL you have, the better you are protected against cardiovascular disease. And that's why we have to eat enough vegetables and fruits, because they will avoid lipid oxidation. And that is also why there is a maximum limit in the intake of saturated fatty acids, because saturated fatty acids will increase the levels of LDL in the blood. So they are hypercholesterolemic. As they increase the cholesterol levels, they increase the risk for cardiovascular disease. And then the nephas, we already discussed, a little bit of fat, but mainly protein. The metabolism is quite well understood. You can see it here. It's the same for the kilomicrons as for the VLDLs. So these particles will circulate in the blood. They will actually release lipids going into the adipose tissue, for example. There's a lipase which is used for that, which is called the lipoprotein lipase. So you see there are already 3 lipases in the humans. The lipase in GI tract for normal digestion, the hormone-sensitive lipase, which is in the adipose tissue for release of lipids from the adipose tissue, and the lipoprotein lipase, which is present in the blood, residing near the wall of the arteries, and this one is releasing the lipids to go either into the adipose tissue or into the lactating breast or other organs. Once the particle passes through the liver, then the apolipoproteins that are physically there can actually recognize the protein or the particle, and that's how the lipids and the particle can physically be absorbed in the liver. If, however, the apolipoprotein is lost, which sometimes happens, then the particle will not be recognized by the liver. It stays circulating in the blood, and then you will produce the so-called LDLs. Because you all the time, by circulation, you lose further lipids, and then the LDLs will finally deposit in the arteries, which can cause coronary artery disease. The same as what we did with the carbohydrates, we can just look at the metabolism. So in case there is no glucose present, then the hormone-sensitive lipase is active. The lipids are released from the adipose tissue in the form of nephas. They can be transported to the muscles to generate energy. They can go to the cortex of the kidneys to produce energy, and they can go to the liver, where they are converted into VLDLs, and from there they go back to the adipose tissue. They can also be used for energy, and in this case they are producing so-called ketone bodies, which are actually an alternative for glucose to go to the brain. And the glycerol, which is released from the adipose tissue, can also be converted by gluconeogenesis into glucose, and that one mainly will also be used by the brain. It seems that the amount of nephas in the blood are very significantly influenced by the mealing patterns, and this influence is very drastical quite quickly after the meal. So for example, at time 0, before time 0, you see that the insulin is low, the dashed line, while the plasma nepha levels, so the presence of lipids in the blood, is very high. As soon as you start eating sugars, within 1 hour, the insulin levels are maximized, and simultaneously the nepha levels go down to almost 0. So you can see here that in 1 hour time, you can immediately switch from lipid metabolism into carbohydrate metabolism. And then as a function of time after the meal, you see that the insulin levels go down again, while the NEFA levels are steadily increasing until they are maximized again, and then they go down. So in this way, you're continuously switching between carbohydrate metabolism and fat metabolism, and the regulation is done by insulin. If you eat a carbohydrate-rich breakfast, then of course insulin is physically present in the blood. You see the kilomicrons that are entering from the lymphatic tissue. A carbohydrate-rich breakfast and a fat-rich breakfast could be, for example, an English type breakfast where you take in bacon and eggs. And this is next to carbohydrates, also an important part, lipids. So all the lipids are now converted into kilomicrons. Via the lymphatic tissue, they end up in the blood, and from there they can go to the adipose tissue. Insulin is released when there is sugar present in the diet, and it means that all the lipids will be stored in the adipose tissue and partly also in the muscles, but mainly the adipose tissue is storing the lipids. There is a specific type of lipids that we already discussed very shortly in the introductory lecture in the beginning of the course, which are the so-called essential fatty acids. And essential fatty acids cannot be produced by humans, so they have to be taken in from the diet. And here you can see the definition. Animals, including humans, do not have enzymes to induce an unsaturated double bond between C9, counting from the carboxyl group, and the terminal methyl group. So in case you can find double bonds in that region of the fatty acid, then you know that it has been derived from an essential fatty acid. And the two real essential fatty acids in humans are linoleic acid, C18,2 omega-6, and linolenic acid, C18,3 omega-3. We discussed these abbreviations in the introductory lecture on biochemistry. So if you do not know anymore what this exactly means, please go to that document and there you will find the explanation. And so 18 stands for the carbons, 2 stands for the unsaturated double bonds, and omega-6 or omega-3 stands for the first position where the first double bond is found in the molecule. Now these compounds are essential. When you take them from the diet, they are converted into long-chain polyunsaturated fatty acids. And starting from linolenic acid, you can produce EPA and DHA. EPA stands for eicosapentaenoic pentanoic acid, and DHA is docosahexaenoic acid. Abbreviations C25 omega-3 and C22,6 omega-3. This conversion is done by enzymes which are called desaturases and elongases. So desaturations means you introduce more double bonds. Elongation means you make the molecule longer. If you start from an omega-3, you can only end up with an omega-3. So it's impossible to start from an omega-6 and to produce a long-chain omega-3. That's not possible. It will always stay an omega-6 once you start with an omega-6. That's why the ratio between the two is so important. In our westernized diets, we consume a lot of omega-6 and not enough omega-3. So it means that we are not producing a lot of EPA and DHA, but we are mainly producing PUFAs that are coming from the omega-6 fatty acids. If we are increasing the consumption of omega-3, we automatically also increase the production of EPA and DHA. And these lipids, EPA and DHA, are found in fish. That's also the reason why it is recommended to consume from time to time a little bit of fish, because these lipids protect against cardiovascular disease. The efficiency of these lipids is rare. It's not so easy because you do not need a lot of them. And so the recommendations are quite low. And even if you take a very tiny, small amount of lipid, for example, by using nuts, it can already be sufficient to supply the recommendations for these essential lipids. Here you can see the interconversion and how it works. So we have three groups of lipids, omega-3 or N3, but N and omega is the same. So N3 fatty acids and 6 fatty acids and then 9 fatty acids. This is the enzymes, desaturases and elongases, and you see how they get longer. And so just going from left to right, you can see in the three families how long-chain polyunsaturated fatty acids are produced. And you cannot switch between the pathways. So if you start from omega-3, or in this case omega-6, you will always end up with omega-6. Here always omega-3, and here always omega-9. If all the three are present, then the body will first start working with omega-3. If omega-3 is absent, then they will start with omega-6. And if both are absent, then they start with omega-9. So the chance that in the body omega-9 metabolism is dominant is very low, because usually we consume on a regular basis omega-6 and omega-3 fatty acids. Why are they important? Well, these fatty acids will be used for the production of hormones. And for example, EPA, eicosapentaenoic acid, will produce hormones of the 3 and the 5 series, while arachidonic acid, which is an omega-6 fatty acid, will produce hormones from the 2 series and the 4 series. So technically speaking, if you look to the formulas, you will see that these hormones are quite similar, but the substitutions is on different locations. In this case, it's on position 3 and 5. In this case, it is on position 2 and 4. So these are very subtle, small differences, but these small differences can already lead to very significant metabolic effects. The best example is blood clotting. If you take an Eskimo, Eskimos are consuming a lot of fish, a lot of marine type products. So in those situations, the supply of EPA, which is rich in marine products, is very high. So it means that Eskimos can produce a lot of hormones from the 3-5 series, but not from the 2-4 series, because their intake of omega-6 is very low, so they do not produce a lot of arachidonic acid. So Eskimos who have a bleeding nose, it's very difficult for them to stop bleeding because the blood clotting is very low due to the high presence of EPA. While in the Western world, where we are living here now, the intake of fish is very low, so the EPA exposure is low, and we are consuming more lipids from arachidonic acid. So in our situation, we have more production of hormones from the 2-4 series than from the 3-5 series. And in our situation, it means that blood clotting is very easy. And more easy blood clotting also means more cardiovascular disease. So it's a clear factor that can influence cardiovascular disease. And that's the reason why you need an optimal proportion between omega-3 and omega-6. And in the westernized society, fatty fish is a little bit stimulated to have more intakes of omega-3. While in Eskimos, technically speaking, it would be better if they have not too much EPAs and that we can correct it a little bit in order to have less bleeding formations. Okay, so the last 5 minutes are actually going to amino acids and proteins. It's only a short part, although I have to say that it's a quite difficult part in human metabolism. And there is quite some attention to it because we now understand that if we can influence amino acid metabolism, we can also influence muscle metabolism. And there are some target groups in our society. Think, for example, about elderly, where muscle metabolism is important. The same, for example, with I would say people that are working in sports and athletes. Also, their muscle metabolism becomes an important issue nowadays. So amino acids can be oxidized, so they're a substrate for generating energy. Normally, the body protein is preserved, so you will not break it down because then you actually have a loss of functionality. And in amino acid metabolism, the most important organs are the skeletal muscle, 40% of the body weight, and the liver. And the liver is actually the organ that receives the amino acids after absorption. It can convert amino acids into carbohydrates, and it synthesizes urea, which is one of the breakdown products if we're using amino acids in the diet. We know that muscles are built up and broken down in a continuous way, and we call that turnover. So a constant cycle of breakdown and building up. And we do that by the use of isotopes. So today we use stable isotopes, which is carbon-13. In the past, they were using carbon-14, which is a radioactive labeled isotope. So what you actually do is you bring the isotope from a certain amino acid into the blood. We know that the loss of the isotope from the blood is directly linked to the rate of muscle synthesis. So the amino acid is incorporated into the muscle, so it leaves the blood, and the speed by which this happens is a measure for the incorporation of amino acids into the tissues, so into the muscles. So what we do in practice is we just infuse it into the blood, and the rate of infusion should be equal to the rate of the synthesis. And those two are a little bit in balance. The amount you have to add to the blood to keep the blood stable is a measure for the amount of amino acids going into the muscle. That's how we can measure uptake of amino acids into the muscle. There is a cycle. So this cycle is a turnover cycle. Proteins that are present in the muscle are broken down, producing amino acids. These amino acids are then used again to produce protein again. So this cycle is the turnover cycle. And the building up is measured by the stable isotope. I already explained that to you. So we can also check the amount that comes in from the diet, the amount which is lost from urea into excretory products. And there is a sort of a balance that keeps the amino acids stable in the blood. And this balance is determined by everything that is arriving in the blood, coming from the diet and coming from the muscles, and everything that is leaving, which is everything that is excreted and everything that goes into the muscle. So this one we already know. Those two we also know. Since this one is in balance, we can also calculate what is coming out of the muscle. Yeah. So what we lose is equal to what we gain. We have already 3 values measured, and we just can measure the unknown value, which is then a value for what comes back from the protein into the blood. And so we can measure this, and we can measure the speed by which proteins are built up and broken down again. And I think I have some values for that. Here you can see that. So if we really look to these values, then you can see that the highest turnover is found in the small intestine. 82% of the small intestine is replaced every day, which is a very active metabolism. So if people are under fat, if no food is actually supplied, then the first thing that happens is that the metabolism of the small intestine will slow down, and it means that these people will gradually lose their microvilli and their villi. So the absorption of nutrients in the small intestine is delayed, or in the worst case, is just lost. So that you need special design foods in order to bring them back to the normal situation. So it's very critical if people are malnourished, especially here near the small intestine, the absorption behavior is being disturbed. We also have the deuterium, which is a heavy water, a stable isotope, which is now used to measure, for example, protein synthesis, fat synthesis, and RNA DNA synthesis. So we can actually measure the speed by which all these processes take place by just following how the stable isotopes are moving through the system. And if you want to know more about it, there's an article online on Ch.4 by Brook et al. That explains a little bit these principles. An important process in the breakdown of muscles is the so-called transaminase. So in this case, you have the transfer of an amino group to an oxo acid. For example, alanine is converted into pyruvate, aspartate into oxaloacetate, and glutamate into 2-oxoglutarate. And you can also go the other way around. So you can make glutamate, aspartate, and alanine by just using these cycles. And it also means that these three amino acids are non-essential because you can produce them from these oxo acids. This brings you to the essential amino acids, and there are three different definitions of it. The most important one is the metabolic approach, which means that we cannot synthesize it with the enzymes we have. Yeah, that's a straightforward definition of an essential amino acid. The second definition is that we do not have the right starting products to produce it, although the cycle is there. So we could produce it in case we have enough supply of starting material, but in case this is absent, then the amino acid becomes essential. This is called the nutritional approach. And the functional approach means that the amino acid plays a role in the physiological functions in the body, and if this is not well produced, to maintain that physiological function. And so this is the most recent definition, but the most classical one is, of course, the metabolic approach. We can't produce it because the enzymes are lacking. And all the rest is non-essential, which can be synthesized mainly from transamination, and in some conditions, amino acids can become essential. We call them then conditionally indispensable or conditionally essential amino acids. For example, cysteine can be produced from methionine, but if methionine is not there, then cysteine becomes essential. The last two slides are showing you the real metabolism. So the first one is in the muscle. So it seems that when amino acids are arriving in the blood, you have a whole chain of amino acids, mainly enriched in branched chains. They enter into the muscle. They can be used to produce muscle protein, and everything which is broken down or which is left over can be converted by two transaminations. So number 2 and number 1. And the final products are two amino acids, alanine and glutamine. So although you get several amino acids coming into the muscle, only two of them will leave the muscle, alanine and glutamine. And these are the two nitrogen transporters that can go to the rest of the body organs. And here you can see that. So when amino acids are entering in the intestine, they are absorbed in the blood. They can go to the liver. The liver will enrich the mixture with branched chain amino acids. They will go to the muscles. And from the muscles, alanine is released or glutamine is released. Alanine can go to the liver for production of glucose. Glutamine can go to the kidneys or to the intestine, where it will produce alanine, and the alanine further goes to the liver. So you get a cycle here, and this cycle is highly influenced by nutrition. If you're well fed, you have a normal cycle. If you're underfed, then there is more amino acid coming from the muscles, meaning that you get muscle wasting.